Good morning, everybody. First, I would like to thank the organizers for inviting me. It's a pleasure to be here today. So I will be talking about the genetic basis of autism, and I'm going to give you an overview of what we have learned so far. I know that there are many families here today, so I'm going to take the time to explain some basic things that I'm sure many of you know, but maybe others don't. So I would like everybody to understand at least the basic messages of this complex aspect of autism. So, as you all know, autism spectrum disorder is a behaviorally defined neurodevelopmental disorder characterized by social and communication deficits and restricted and repetitive behaviors. It has an early onset, usually before the first 3 years of life, a strong male bias, and it has a prevalence of about 1%. It is marked by, it's characterized by marked clinical heterogeneity, with intellectual disability in about half of the individuals. There are others that have normal intelligence, and even a minority that has very high IQ. About a third of patients have epilepsy. Some patients have absent language, and there is also a proportion of patients that, in addition to autism, have other associated features, such as dysmorphic features. These are small or big malformations, for example, of some facial features, or the hands, toes, or even internal organs, the heart, the kidneys. And when there are these associated malformations, we refer to this type of autism as syndromic autism. So we know that there is a strong contribution of genetic factors in ASD, and we know this mainly because of familial studies. When we study the recurrence risk in siblings, this is what's the chance of having a second child that is affected with autism when you already have one child that is affected with autism. We see that the recurrence risk is way higher in families with one affected child compared to the general population. So the risk, it's 10 or 20%, more like 20%, so 1 in 5 chances of having a second child with autism compared to the general population. And then twin studies told us that indeed there is a strong contribution of genetic factors. So we compare the concordance rate. This is what's the chance of the second twin being affected with autism when the first one has autism. And we compare monozygotic twins. These are identical twins that share all of their genetic information. We compare them to fraternal twins or dizygotic twins who only share 50% of the genes. And we see that the concordance rate is higher in monozygotic twins, so it's 80–90% compared to 10 or 20% in dizygotic twins. The fact that the concordance rate in monozygotic twins who share 100% of their genetic information is not 100% means that not everything in autism is genetic. There are some other factors that have to be non-genetic. If everything were explained by genetics, the concordance rate would be 100% in monozygotic twins. So, when we use this data in families and twin studies to calculate what's the contribution of genetic factors in autism, we come to estimate of about 50–80%. This means the heritability or the contribution of genetic factors is about 50–80%. So there is the remaining that it's caused by non-genetic factors. And these genetic factors, we don't know exactly what they are, but it could include environmental factors, which we have not identified yet. They could include epigenetic factors or even chance. So stochastic factors, chance can determine whether you get autism or not in certain circumstances. So in conclusion, because there are genetic and non-genetic factors, this means that autism is what we call a complex disorder that has both common and rare variants. As Joe said, much of the progress, or I would say all of the progress, in our knowledge of the genetic of autism in the past years has come from the study of rare variants. So I'm going to focus my talk on rare variants. So today, we can identify a specific genetic defect in about 20 or 30% of patients with ASD, okay? And what are the types of genetic defects that we can find in these individuals? We can find chromosomal abnormalities. We can find monogenic disorders. This is disorders that are caused by a mutation in a single gene. Or we can find what we call copy number variants, or CNVs. CNVs are abnormal copy numbers of a section of the DNA. So we normally have 2 copies of every chromosome, one inherited from the mom and one inherited from the dad. If we're missing one of these copies of these segments, it's called a deletion. If we have an extra copy, so instead of 2, we have 3, it's called a duplication. So I'm going to use these terms, deletion and duplication, quite often. And the global term to refer to these deletions and duplications is CNVs, so copy number variants. So I'm going to explain in detail what are these type of genetic abnormalities, but just to give you an overview, what we know today is that all the causes of autism are very rare. It means that when you take a group of 100 individuals with ASD, probably you're not going to find the same genetic cause in 2 of them, okay? Each of them is going to have a different genetic cause. The most common cause of autism is Fragile X syndrome, which is present in about 2% of patients. There is a major contribution of de novo variants. The de novo variants are the ones that appear spontaneously in the kid and are absent from both parents. So even though we know that de novo variants play a very important role, we've also learned more recently that inherited variants can also contribute. And in most cases, even though they are inherited from a parent, the parent does not have autism, okay? I'll come back to this concept later. And what we have also seen very clearly during the past years is that there is a considerable genetic overlap between the genetic causes of autism and those of other neurodevelopmental disorders, such as intellectual disability, epilepsy, schizophrenia. Okay, so I'm going to give you an overview of some of the terms that I just used so you understand better what I'm going to explain afterwards. So in every cell of our body, we have 23 pairs of chromosomes. These pairs are numbered from 1 to 22, and there is one pair that is called the sexual chromosomes. So in every cell, in the nucleus, there are all these chromosomes. Together, we refer to this genetic material as genome. The chromosomes are formed by the DNA, and the DNA contains the genetic information. And in the DNA, there are genes, and the genes contain the instructions to produce proteins. And the proteins form all of the body and are involved in important functions. So when there is a defect in a gene, it's like there is a defect in the instructions to produce a protein. The protein is going to be either absent or defective, and thus the function is not going to be done correctly. So if we compare the genome, which is the whole of the genetic material, to a book that would be called the human genome, then the genome or the book has 22 pairs of chromosomes, 23 pairs of chromosomes. This would be a book that has 23 chapters. So when we say that there is a defect in a chromosome, it's a quite big piece. So it could be like you have a defect of a whole chapter in a book, either one chapter that is missing or one chapter that is repeated and it's printed in double. The chromosome contains many, many genes. This would be equivalent to sentences in a chapter. And the genes are formed by nucleotides or letters. There are only 4 nucleotides that contain all the instructions of the human genome, and so they are the equivalent of letters. So if we use this analogy to explain the chromosomal abnormalities that are defined in autism, we can find effects touching the chromosomes. This could be abnormalities in the number of chromosomes. You can have a loss of one chromosome or have an extra copy. When you have an extra copy, this is a trisomy. For example, the patients that have Down syndrome, they have 3 copies of chromosome 21, and this is the cause of their difficulties. Or you can also have structural abnormalities in the chromosome, and these structural abnormalities are very varied, but as you can see, they can involve a deletion, so a big chunk of chromosome that is missing, or a duplication to 1 extra copy of this part, an inversion, translocation with the tips of 2 different chromosomes are switched, or for example, a ring chromosome, where the extremities of the chromosome are deleted and the chromosome closes itself and forms a ring. All these are chromosomal abnormalities, and about 3 to 5% of kids with autism have a chromosomal abnormality, one of these types, that explains their disorder. Okay. We can also find defects in the genes, and the defects in the genes could be dosage defects. So as I said, either you have a missing copy of the gene, or you have an extra copy of the gene, and both of these are deleterious and can cause problems. Or the defects in the gene can happen at the nucleotide level, so the letters, so not just a big chunk of DNA that is abnormal, but just one of the letters in the instructions of the gene that it's abnormal. So instead of having a G, you have an A, and all the instructions go bad, and you're not going to be able to produce the protein correctly. So to recapitulate, there are 2 main types of genetic defects. One is chromosomal abnormalities, including CNVs, that affect thousands or millions of nucleotides. And the other is DNA sequence mutations. We also call these point mutations, where there is only 1 single or a few letters that are affected. Okay. And there are 2 non-exclusive mechanisms that we think can result in autism. So the main one is we have a gene, there is a defect in the gene, as I said, deletion, duplication, or a sequence mutation that produces an altered protein that results in a biological effect and results in ASD. So this model, it's what applies to rare variants, okay? These rare variants have a strong effect, meaning if you have a mutation in one of these genes, for sure you're going to be severely affected, okay? So of course, they're absent from the general population, meaning unaffected people cannot have mutations in these genes. And because a single variant is enough to cause a disorder, we call these Mendelian disorders. The other model, which we think participates in autism but has not been demonstrated, it's the common variant weak effect. This model says that there is not a strong defect in only one gene, but there are small, tiny defects in many, many genes. So if you have, for example, and we don't know how many genes, I just drew 3, but we don't know how many, but let's say that you need a defect in 100 genes to have autism. So if you have a defect in 20 genes, small defects, you're not going to be affected. But if dad has defects in 50% and mom has defects in 80% and the kid inherited 100 defects, small defects, he is going to be affected with autism, okay? So these variants, because they have weak effects, they can be present in the general population, but when they cross a number that we don't know yet what it is, then you become affected, okay? So this is not caused by a single gene, but by an interaction of many genes. So these common variants, as I said, I just explained this model. We think that this explains a proportion of autism. We don't know how much. I'm going to move back to rare variants. we know today that rare variants explain 30% of cases of autism because this is the cases where we have found that there is a rare variant. We don't know if in these cases when we identify a rare variant, there is also a contribution of this common variant mechanism. We don't know that yet. And we don't know what proportion we're going to be able to explain with the rare variant model. Okay, so what strategies do we use to identify the genes involved in autism concerning rare defects? So the first one, to identify chromosomal abnormalities, is to do a karyotype. And the other is to search for deletions and duplications. These techniques have evolved during the years. So at the beginning, many times ago, we used a technique that was called FISH, then a technique that was called MLPA, and now what we use is one that's called microarrays because it allows us to look for deletions and duplications in whole genome. So we can study with only one test all the chromosomes and search for deletions and duplications in everywhere, okay? So there is a recommendation today that all the kids affected with intellectual disability and affected with autism should have a microarray. Microarrays identify an abnormality in about 10% of kids with intellectual disability. But in autism, the etiologic yield, so the number of patients that are diagnosed with microarrays, is lower, and it depends on whether, for example, they have syndromic or non-syndromic autism. Kids that have isolated autism without any malformations are less likely to have one of these rare abnormalities, okay? And those, for example, that have severe intellectual disability are more likely to have one of these abnormalities compared to those that have no intellectual disability. Okay. And the other technique that we use to identify rare defects in autism is search for sequence mutation using sequencing. And there are different techniques to sequencing, but the one that is preferred today is called whole exome sequencing, which allows us to sequence simultaneously all the genes in the genome. So it's very powerful. And it could be the exome, which is the part of the genes that contain the instructions to produce the proteins, or it could be the whole genome. The whole genome, it's much more expensive. It gives more information, but if you compare the yield of diagnosis with the cost, today the preferred technique is whole exome. So there are over 100 genetic defects and genomic disorders that are associated with autism. Probably there are many more. We're going to continue identifying them. This is just a few of them. You're not supposed to read these. This is just to give you an idea of the sheer number of genetic defects that can result in autism. As I said, the most common one is Fragile X syndrome, but there are others that you have probably heard of: Rett syndrome, tuberous sclerosis, P10 autism, macrocephaly syndrome, or, for example, Phelan-McDermid syndrome caused by deletions and mutations of the gene CHANK3. Okay. And what is interesting is that all these disorders can cause autism, but all these disorders are also well-known causes of intellectual disability. So this shows that at the genetic level, the causes of autism and intellectual disability are the same, okay? But in some patients, these defects, in some patients, they can manifest as intellectual disability alone. In some patients, they manifest as intellectual disability with autism, and more rare, in some patients, they can manifest as autism without intellectual disability. And we don't know why it is that sometimes the same exact mutation can manifest in one patient, intellectual disability with autism, autism alone, or autism without intellectual disability. Okay. There is also a marked overlap with the causes of epilepsy. So again, you're not supposed to read this list. It's to show you that all these genes and genetic defects are causes of epilepsy, and all of these can also be found in some patients with autism, okay? So a marked overlap with epilepsy, and which is interesting is even though some of these genes are called epilepsy genes, sometimes you find mutations of these genes in patients that do not have epilepsy. They can have intellectual disability alone, or autism alone, or both without epilepsy or epilepsy. So what we see is that there is variable expressivity of genetic defects. This is the term we use. The expression, the clinical manifestations of these genetic mutations can vary from one patient to the other. Variable expressivity. Again, we don't understand the mechanism yet. This is something that we have to keep working. It's going to be very difficult to understand, but we hope that one day we're going to understand it better. Okay. So today, we know that there are at least 190 genes that have been clearly involved in ASD, all through rare variants. But these genes, as I said, only explain about 20 or 30% of cases, meaning that we still have a lot of genes to identify. And we calculate that maybe there are going to be about 1,000 genes that are involved in ASD. So we still have a lot of work to do to identify the remaining genes. And as you see, these arrows indicate where we started to use microarrays in ASD in 2009, and when we started to use whole exome sequencing in 2012 in ASD, and you see that the progression in the identification of genes is mainly due to the use of these genome-wide techniques in very large samples. So, if we put together this information that I have showed you before, and even quite recently, people would use to think of autism as all the patients have autism, behaviorally defined defects, and because they have a behavior defect that is similar, this means that the genetic defect underlying these behavioral difficulties must be the same. This is the blue model on the left. You should completely forget this model because this is absolutely not how it works. If you have a group of patients with autism, it's most likely that even though the behavioral manifestations are similar, they all have different underlying genetic defects. So as I said, most of the progress has come from the study of CNVs, copy number variants, and sequence variants. So one of the studies that contributed to the understanding of the role of CNVs in ASD is that produced by the Autism Genome Project, where we participated. The Mount Sinai group also played a very active role in this consortium. And we searched for these CNVs genome-wide using high-resolution arrays in over 2,000 families. And we published the results in 2010 and then in 2014. And what we showed is that individuals affected with autism have an enrichment of rare CNVs, and that CNVs play a major contribution in the onset of autism. And most of these CNVs are de novo, meaning they have a very strong effect, they are absent from the parents, and they appear spontaneously in the kid. But also inherited CNVs contribute. And we identified for the first time several genes involved for the first time in ASD. So again, this list, you're not supposed to read it. This is to show you that among these over 2,000 patients with autism, we identified in 82 individuals 38 different causes of autism. So we made a diagnosis in 3% of the sample, and this sheer number of different causes of autism shows you the diversity. So some were recurrent. For example, there is a gene that is called neurexin, and there were 8 patients that had neurexin deletions. So 8 among 2,000, okay? But there were many others that we found only once, okay? So most of the things are not recurrent. So as I said, most of these pathogenic CNVs, so the word pathogenic means that we interpreted clinically these CNVs, and we think that this CNV is causing a deleterious effect. We call them pathogenic, and we think they are responsible for the phenotype, so for autism in this case. So most of the deleterious ones were autism, but as you can see, in 36%, they were inherited from a parent. And in most of the cases, the parents were not affected with autism. This means that these CMVs are associated with what we call incomplete penetrance or variable expressivity. So incomplete penetrance means that a genetic defect can cause problems in some patients, but not always. Sometimes, even if rare, it can happen that some of the carriers are not affected. We don't know why. We don't know why this CMV, sometimes it causes a very severe form of autism, and then the dad is an engineer who works, got married, and has no main problems, okay? Sometimes they can cause what I refer to as variable expressivity. So even though the carrier parent does not have autism, maybe they have problems learning to read, or problems, some types of social problem that don't qualify as ASD, but can interfere with their life, or they have depression, or they have bipolar disorder. And we think that maybe there are some alternative manifestations of these CNVs. Okay, and when we look at these pathogenic CMVs, we see that they are not always associated with intellectual disability. So even though all these CMVs that we identify are known causes of intellectual disability, as I said, not in all patients they are associated with intellectual disability. And although many of these CMVs are well-known causes of syndromic autism, remember the form of autism that is associated with abnormal facial features or malformations, in 83% of these cases, they were associated with non-syndromic autism. So to give two contrasting examples of the type of CMVs that we identify of autism, the first one is Phelan-McDermid syndrome, that you're going to hear more about this in the next talk. So Phelan-McDermid syndrome is caused by a deletion in chromosome 22, or that involves the gene CHANK3. And okay, so you see here that we identified 4 patients with deletions. The deletions are drawn by these red lines. So all these 4 deletions involve the gene CHANK3. And you see that there are some small deletions that only involve this gene, and there are others that are bigger that also involve other genes. The genes are here drawn in blue, okay? So among over 2,000 patients, we identified 4 deletions that involve the gene CHANK3. It means that these 4 patients have Phelan-McDermid syndrome. All the deletions were de novo, because Phelan-McDermid is a syndrome that is characterized by complete penetrance, meaning if you have a deletion or a serious genetic defect of CHANK3, you're going to be affected for sure, and severely affected, okay? So when I said that there are some CMVs that are associated with incomplete penetrance or variable expressivity, that does not apply to every single CMV. It applies only to a subgroup of them, okay? Phelan-McDermid syndrome is one of the most frequent causes of ASD and intellectual disability. We think that it can explain about 1% of cases. And another completely different example of the CMVs that can be identified in autism is this CMV that it's called 16p112 deletion or duplication syndrome. So we can find deletions drawn here in red. So each red line is the deletion in a patient. Or we can find duplication. So these are missing 1 copy, and these have an extra copy of this same region. And you see that almost all the deletions, and not almost all the deletions are duplications, have very similar breakpoints. You see the size is very similar. This is what we call recurrent CMVs. And so they all affect the same number of genes. There are over 20 genes in this interval, and we don't know which of these genes is responsible for the phenotype. But what we do know is that deletions are found in patients with autism, with intellectual disability, and even with obesity. but obesity with intellectual disability and autism. Whereas duplications are also found in autism, intellectual disability, but also in patients with schizophrenia and in patients with epilepsy. And whereas deletions are associated with increased body weight, duplications are associated with decreased body weight. And this is a typical CMV that is associated with variable expressivity, because as you see, one carrier can have schizophrenia or epilepsy or autism or intellectual disability, which is something very, very different. And you can imagine that at the beginning we were very, very surprised to see that such different phenotypes could be caused by the same single CMV, okay? And again, we don't know why it is that the same exact CNV affecting the same exact number of genes manifest with schizophrenia in this patient and with epilepsy in the other one. Okay? Deletions and duplications in this region are associated with autism only in about 20% of cases. And as I said, they're also involved in other neurodevelopmental disorders. So this is a typical example of a CNV associated with variable expressivity. So I said that main contribution in the field came from the CNV studies, but the most recent contribution, a huge advance, came by being able to sequence at the same time all the genes in the genome using whole exome sequencing. So the first studies started in 2012, and they continued in 2014. Again, here, the Seaver Center and the Mount Sinai group played a very, very important role in these studies. And overall, these studies have studied about over 4,000 families with ASD. The analysis have focused on the role of de novo variants, and as I said, inherited variants also play a role, but they started to analyze, let's say, the most evident or the most easy thing to analyze, so they analyzed mostly the de novo variants. So they showed that they play a very important role in the etiology of autism. These studies also helped to identify many genes that were involved in autism because there were recurrent de novo mutations in these genes found in patients with ASD. And these studies also helped to show that many of the genes that were mutated in ASD were also found to be mutated in exome studies in intellectual disability, in epilepsy, and schizophrenia. So again, this idea of overlap. Okay. And another thing we had suspected before, but was shown clearly with these whole exome studies, is that older fathers transmit more de novo mutations to kids. Okay? And when you look at this slide, this includes data on autism, but also on schizophrenia, because it's the same thing for schizophrenia. Here, it's the age of the fathers, and you see as the age of the fathers increase, the number of de novo mutations increases. So it's not starting at what age the risk increases. You see that very early the risk starts to increase, okay? So most de novo mutations are paternal in origin, and they increase with the age of the father. So I've been saying all these genes have a lot of functions. I mean, there are many genes involved in autism, but what are the functions of these genes? So they are involved in a myriad biological pathways, but they converge on certain common pathways, such as synaptic genes, the genes that served in the communication of the neuronal cells, or in the regulation of other genes. So these are the synapses, the communication of two nervous cells, and these synapses are the basis of everything we do with our brain. We think, we talk, we feel, everything with the synapses. When the synapses are not functioning well, they're not transmitting the messages well, there are going to be problems. These are many different genes in the synapse that have been involved in ASD. And what's the importance of identifying the genetic cause of autism? I think this is an important message. There are benefits to the patient, like, for example, if you identify the specific genetic cause, you can give informed care, for example, anticipate and manage medical comorbidities. and they can participate in clinical trials when available. This is like the beginning of personalized medicine. There are also benefits to the family. First, it's the knowledge about the cause of the disorder. They can have an answer and stop worrying about what might be the cause. They can get information about the clinical course of the disorder, specific information, because if you don't know what's the specific cause, you cannot tell them exactly how is it going to progress. They can get genetic counseling, so if you know exactly what's the genetic defect, you can tell them you have a recurrence risk for the other kid that it's maybe 1% or maybe it's 20% or 50%. And the families can participate in family and advocacy groups. And to society, there are many benefits also, like clinical information on a population level. We identify new targets for medicine, develop model systems like mouse models that we're going to hear afterwards. We understand the mechanisms of the disorder and develop novel therapeutics. So in conclusion, there are many genetic disorders that are associated with ASD. All the genetic defects identified thus far are very rare. De novo variants as well as inherited variants play an important role. All individuals with ASD should be explored to try to identify the genetic etiology. The current diagnostic yield, 20 or 30%, indicates that hundreds of genes remains to be identified. And the identification of genes involved in ASD illuminates the neurobiology of the disorder and provides targets for novel therapeutic interventions. Thank you.